So I'm Dave Sands. I'm a plant pathologist. I like to consider myself sort of hardcore science. We try to do experiments that have never been done by anybody. And the problem with that is once you do them and once you're successful, and they may have world impact, most professors don't know how to handle all the things that have to happen after you have a successful science. So basically, I started working on diseases of plants maybe 50 years ago. And like any plant pathologist, I work on diseases of plants. Some of the plants are weeds, some of the plants are crops. Almost 99% of all the plant pathologists have a job where they have to protect plants, crops. But we always look and find diseased weeds. For instance, this is a dead Canada thistle. Why is it dead? It has vascular invasion by a fungus. And that fungus, if it's fusarium, only attacks one weed. So, why aren't fungi used to control weeds? Because you can only use a fungus if it attacks only one weed. It can't attack Mrs. Smith's zinnias or somebody's tomatoes. It has to be specific. Secondly, why aren't bioherbicides used? Because we have chemicals that kill everything. If it's green, they kill it. If you're a farmer and you have 10 weeds, imagine having to have 10 fungi. But there are hundreds of plant pathologists, and with the problems that we now have with the chemical industry, I wish we'd been pushing farther ahead. There were hundreds of papers on diseases of weeds and hundreds of grants, but we have no commercial products out where you can say, I'm going to use this fungus to kill spotted knapweed or Canada thistle. And yet we're in dire need of that. And we will explain how we developed this technology and it actually works. First, about 30 years ago, this government guy walked into the lab and he said, gee, you can kill weeds? I have some weeds I want you to kill. Cannabis, opium poppy. This was the war on drugs at the time. And so he gave us some money and then some more money and said, get good at it. And after 10 years, we got good at it. Next slide. If you look on the right there, you see some full-grown cocaine plants. And if you look at the left, you see the cemetery. We did that. We did that with a fungus named Fusarium oxysporum. And it was, at the time, we thought, much better than spraying herbicides all over South America, killing everything. So we had host-specific fungi for opium, for cannabis, for cocaine. And the big problem then was, how do you get it out and deliver it? And we found a way to fly C-130 airplane at night at 3 in the morning and drop down seeds that had fungi on them. And the fungi would go down the root system. And that was what we were paid to do, and that's what we did. Next slide. And you might say that although we got very good at it, these systems weren't used. But the National Academy of the Sciences in Washington, D.C. That's kind of a big deal. They published a whole book on the feasibility of using these fungi to kill weeds. Yeah, we got good at it, and it was dangerous. And you can imagine how dangerous it was. There's a lot of money involved in drug crops. Next slide. So here's the problem if you're a plant pathologist and you say, well, I see a diseased weed like the one I showed you here. The problem with a weed disease is a perfectly good fungus doesn't want to kill maybe more than 40%. If you killed your only host, where would you go the next year? So fungi maybe only kill up to 40%. This was an experiment we did on San Clemente Island, and that's a rust fungus. And for controlling wild oats on that island, it was too little and too late. Next slide, you can see that when you had no rust, over 60 years, we ran a computer simulation. You got good growth of the wild oats, but even when you had the rust, you always had 40, 50% of the wild oats living. So this was a problem. Fungi, next slide, don't generally want to kill. They want to take something like a real estate agent or something, a brokerage fee, but if they kill their only host, they're out of luck. So this is where we departed from all other plant pathologists, in that we read the literature that Steinberg in 1960 found that there was a disease called Frenching disease that had been around for 200 years on tobacco, a big cash crop for 200 years. And sometimes the seedlings looked like that on the right. And what he found is that these were simple soil bacteria producing an amino acid. What? An amino acid? The same thing that you use in your diet? The things that you need to make protein were toxic? And he found that they were. And so you don't need glyphosate. You don't need imazapyr. These are made in big chemical companies. They're called synthetic chemicals. You could use amino acids. And the beauty of amino acids is all living organisms have to make amino acids, although animals can't make 9 or 10 of them. They can make another 10. Plants can make them all. Fungi can make them all. And that was our break. We said, well, if amino acids will kill a plant, why get complicated? Next slide. So we brushed up on something called biochemistry, intermediary metabolism, and we figured out from the literature how amino acids are made. That lysine, methionine, threonine, isoleucine, they're all amino acids. Homoserine is too, asparagine is too. And how are they made in the cell? They're made the same way in basically all cells, more or less. And they have a top enzyme. And that enzyme, those 3 colored arrows up there, are each isozymes. They do the same thing. They take aspartate and they move it to aspartyl phosphate by adding a phosphate. And then downstream you get all this other stuff. But plants have their own internal environment. So plants have a weakness, an Achilles heel. They have lost 2 of the 3 enzymes up there. So where lysine regulates one, methionine another, and threonine another, they usually have trimmed it down where they even only have one of those enzymes. So if you add lysine to a plant, it might kill it because it'll shut down the whole pathway and the plant will starve and not be able to make protein. So by understanding biochemistry, at least a little bit, for a plant pathologist, most people in biochem class that are plant pathologists get a deer in the headlight look, but that's not good because this is basic to all living systems. Next slide. So if we think about how we could solve this problem, especially in countries like Africa or even Montana, do we really want a big chemical factory? Or can we get a host-specific fungus to simply produce the amino acid as it attacks a plant? And instead of killing 40%, it kills 95%? Maybe that is a way to go. You don't pollute the water. You don't have a big chemical plant. It's kind of bottoms-up science. And so how do we do that? It's easy. There's 200 amino acid analogs that look like amino acids, but they have a different methyl group or something on them, or they have selenium instead of sulfur, something. And if you put an amino acid analog in the middle of a plate, let's say for valine, the analog is penicillamine, you kill all the fungi spread on the plate near the zone. But if you look closely, which you can't see here, you will find a few mutants that are overproducing valine, and they live. So that's how we select for amino acid excretors. Once we have an excretor, what do we do with it? We have to make sure it excretes. So we have an agar with a bacterium in it that requires valine. And it will only grow if there's valine present. And you can see our mutant on the left is making a lot of valine, and a wild type normal fungus pathogen doesn't make a lot of valine. It makes a little bit, but not nearly as much, like 100 times less than the one on the left. So we can get fungi to make amino acids. We can have our factory right in the plant. So all we have to do is figure out a way to get them out. But let's see what happens here. Next slide. Oh, all weeds, in fact, all plants are inhibited by one or more amino acids. So that gives us a huge tool chest. Okay, it's a little bit harder with a parasitic plant because a parasitic plant attacks a regular plant. So you have to make sure your amino acids don't inhibit like corn, but they do inhibit a parasite, a parasitic plant. Those bottom two are parasitic plants there. Next slide. So we tried this out on one of Montana's weeds called hound's tongue. And on the left, you see a wild type bacterium attacking, and it causes kind of like pimples on the plant, nothing serious. But when we get it to make valine, look what happens. So this is our poster child, hound's tongue, and we used a fungus to kill those plants, or at least to greatly reduce them down below their what we call economic threshold, so we can live with that weed. Next slide. So at that point, we quit the government work and we went to Africa. And with very little funding, over 10 years, we worked on their worst weed. Why is it their worst weed? Because a farmer in Africa, a subsistence farmer, may have no more land than what's in this room, and they grow their crop, and if their crop doesn't grow, they starve to death. That's reality. And that little pretty flower there attacking maize has millions of seeds in the soil. It just waits for you to plant maize, or corn, we call it, or sorghum, or millet, all related plants that are all staple food for Africa. So we're working on Africa, where chemical companies don't really do much because you can't make a profit in Africa for lots of reasons, especially with a farmer that only has land this size. So we got our fungus that attacks striga. We isolated a bunch of them. They were at best 40%ers. And then we got them to make tyrosine and leucine and methionine. Tyrosine and leucine are two amino acids that really inhibit striga but don't inhibit maize. And then methionine, the fungus in the soil, if it produces methionine, it causes all the striga seed to germinate. And it's not enough to just kill striga. You want to kill the seed as well. And with this triple threat, or triple way to go, we then put out some field trials. Next slide. And Claire's going to talk about that. Claire is my daughter. This is a university. They don't like nepotism. So they don't want my daughter working with me, but we have world impact here. So with true grit, we proceeded. And Claire's going to talk about it. And the question is, how do you get the fungus out? And the answer is a toothpick. Claire, could you take it from there?
Sure. Hi, I'm Claire Sands-Baker, his daughter. I've been working in nonprofit and non-governmental organization development for 25 years. So about, I guess, 6 years ago, he and I started working on this project together because my skills fit where his skills had led him to. So it was a good partnership. He skipped a couple slides here. This is just a continuation of this research that's going on right now through MSU. And this is the bulk of it here with Canada thistle and field bindweed through some funding from Montana Organic Association, and hopefully funding from the USDA that's sitting in the office in DC waiting for approval. And they've also been working on using a wheat seed to disperse this for the Montana project. All right, so our project's called the Toothpick Project, and we'll kind of fill you in on why it has that weird name. As my dad was saying, a smallholder farmer, a shareholder farmer, a subsistence farmer, these are farmers in Africa, Southeast Asia, Central America. They generally are farming for the purpose of feeding their family, possibly some extra food harvested that they can sell. Patricia is an example of a smallholder farmer that I met in Kenya a couple years ago. She has about an acre of land. She grows maize, maybe a couple other crops. When I was on her farm, I didn't actually see any mango or avocado trees. She has 6 kids. She does the bulk of the work. Her husband, she didn't say why he doesn't help as much, but this is typical in Kenya. 85% of maize farmers, which is the primary crop there, are women. And this is Patricia's field. Those purple flowers are the striga. You can see the maize stalks, corn stalks. And that striga is attacking the roots of the maize plant and zapping it of all of its nutrients prior to even coming above ground. You can see it's pretty devastated, and typically a farmer might lose 20 to 100% of her crop to striga. It's considered the #1 pest threat to food security in Africa, and all of these red countries, those are where it's heavily infested, and it's on 40 million smallholder farms. So if you want to put this into perspective, how many people this is impacting, Patricia has 8 people that she feeds off of her farm. If that's typical 40 million farms, that's 320 million people that are impacted by food coming off of striga, or impacted by striga. And the population of the United States is 324 million. So we're talking about huge, huge impact in food security. They estimate maybe $9 billion in crop loss annually, and it's kind of hard to figure out what that looks like. It's all coming off of the plates of smallholder farmers. This isn't a big industry issue. It attacks maize, sorghum, millet, dryland rice, sugarcane, cowpea, and there's now reporting of it coming off of attacking wheat in Sudan. This is just one country, Kenya, in western Kenya, which is where Striga is heavy. They say it's impacting every single field. We've received reports that it impacts 50% of arable land in Tanzania. Every single country seems to have reports like this. And as I said, it's a food security issue that's gender oriented. There are a couple current interventions. Weeding, fertilizer, push-pull is one where they can plant another plant that draws the striga to it so it doesn't harm the crop. Those are all decent approaches, but they're not restoring the crop yield. Like my dad said, each striga plant can deposit between 50,000 and 500,000 seeds in the soil every season. And those seeds can just sit there dormant until you plant the crop that's going to have the hormones that cause the striga seeds to germinate. So if you're not attacking the seed bank in the soil, you're going to approach this every single year. There are some commercial products. IR maize is a maize seed that was developed to be resistant to imazapyr, which is a chemical. This is similar to the chemical resistant seeds that we're using in the US, like Roundup Ready products. This was not developed for smallholder farmers. Smallholder farmers plant with their hands, and so they decided, oh, well, we'll just give the farmers some gloves and they can plant this chemical coated seed with the gloves, and hasn't actually been very well accepted by farmers. Striga resistant seeds are a great option, but they don't restore a crop yield. I think the highest report I've seen is about 22% increase in crop yield in Striga fields using Striga resistant seeds. And then there's a product called Foxy 2, which he mentioned. The name of the Fusarium oxysporum, we call it Foxy. That's the same technique that they were looking at. They're like, we'll take this fungus that we're finding in the fields and then we'll put it back in the fields and we'll kill the weed with it. But they weren't using the virulence enhancement amino acid excretion selection. So they were just putting the same thing back in, and they basically got that 40% kill of the striga, not high enough to make an impact for the farmers. So this is a paper actually put out by the researchers who failed, which is unusual. A lot of times you won't see a paper come out about failure, but they published a paper saying it didn't do what they were hoping it would do. So here's striga. On the left is the healthy striga plant. On the right, you can see it's diseased. And so that's what our researchers are looking for. Capture that diseased striga seed and then isolate the fusarium off of that that's causing that wilt. And this is just to go over again what he said. Sorry, is this buzzing a lot? Sounds like it, but move it away. There are 200 different forms of of Foxy approximately. And each of them target a different host plant. And so this is really a great key point because it means it's not going to impact other crops. But as he said, the native Foxy isn't strong enough. And so we selected for the tyrosine, leucine, and methionine. There's also that other slide he showed threonine, and that causes harm to maize. So that one was thrown out because we didn't want it to harm the crop and the striga. We only wanted to harm the striga. So we found our excretors there, and then we had to figure out how to get it out to the farmers. So this is a photo from our lab in Kenya. You can see the 3 different colored toothpicks represent each of the 3 different amino acids that are being excreted. And you can put the toothpicks on those petri dishes and the fungi will just grow right into the wood. And then you can dry the toothpicks and put them in a sterile drinking straw. And that can get delivered to the village level, to the farmer. And she can then grow her own inoculum. So here's our deployment. So you take the toothpicks. We're actually using wooden pegs now just because of our fermenter system as we're commercializing. You can take a pot of rice, cook it, let it cool down, and put the toothpicks in there and seal it up and shake it for 3 days. And after 3 days, you just have to shake it a couple times a day. You don't have to shake it the whole time. And then after a few days, all of that rice will be inoculated with the fusarium. And then the farmer just puts a pinch of it, 1.5 to 2 grams with each maize seed. And this is the whole process, the entire process. You can see in this photo, in the center, that's where we used the Foxy. On the left is where we used hybrid seed and fertilizer. Those were the same hybrid seed and fertilizer in both the left and the center. On the right, that was the farmers recycling their seed from the previous year, which is the farmer traditional practice. It's a little hard to tell in this photo how much better that center section is. You can see it's better. This is how much better it is. We saw better yield in all of the fields using Foxy, and the average yield increase was 56% in the long rain season and 42% in the short rain season. And in this photo, you can see the maize cobs. The shriveled ones are ones that had the Foxy in the field, and the larger ones are ones from using our treatment. So this was a trial on 500 farms over 2 seasons. It's a big trial. It was funded by the Bill and Melinda Gates Foundation. My dad got that grant. It was the first Gates Foundation grant in Montana. Kind of a big deal. It's nice to look at the statistics and the numbers, but it's also nice to hear from the farmers how much they appreciate the work and that this effort for them really pays out. These are the implementers. We had 27 women implementers. I got a few men in there. Who each had about 20 farmers that they worked with to help train them on how to manufacture their rice inoculum. Then after those successful trials, we had to start figuring out, okay, now we need to register this, commercialize it. In Kenya, this is the step, these are the steps you have to go through, about 15 steps for registration, and we've been working on it for 5 years now. It takes a really long time, and it's not cheap. And there are also errors that come into play. But this is about typical for what every country in Africa is going. We're going to have to do every country separately. Africa has, that's how the regulation system goes. They don't have like an EU where we just go to Europe and register. We have to do it in every country individually. Here are some of the challenges. We had fall armyworm infestation, which was unexpected. Some of you, if you know anything about agriculture, fall armyworm hit a few years ago, and it actually hit on 4 different continents maybe, and it was pretty devastating. And then we had we have to use third-party implementers, and some of them were negligent, not putting fertilizer on the control, that kind of thing. And then we had drought, and then we had a third-party implementer not even turn in their report. So all sorts of challenges, but right now, these are the results that we have turned into the pest control board. And you can see, we tried a variety of different things. So you can see 1 gram of rice, 2 grams of rice, 3 grams of rice, but rice is actually expensive. It's one of the cheapest things you guys can eat, but it's actually pretty expensive for a smallholder farmer to access and use. So we were trying to find something else that we could grow the Foxy on. And so we used gridded maize cob. So you can see here we also did different doses. So this is a test of doses and substrates to see what works the best. And 2 grams of rice looks like it's the best. We're actually, economically, we've crunched the numbers and we're going to use 1.5 grams of rice. And then all of the results are better than the control, which is that C over there. Another part of registration is to do tox and ecotox studies. And so we did this at Virginia Tech and University of Nairobi, and we didn't find any toxins that are associated with Fusaria. Then we have to not just figure out how a farmer can apply it in her field, but how do we actually get it to the farmer? So we've, it's a public-private partnership. If any of you end up going into commercialization outside of the US, you'll hear a lot about public-private partnership. CALRO is the Kenya Ag and Livestock Research Organization. It's equivalent to our USDA. And we have a manufacturing lab, and so they're manufacturing our toothpicks, our wooden dowels. They transfer them to us, and then we have partners which are in the NGO world, non-governmental organizations, and community-based organizations. Almost every farmer is part of a farmer group with another 15 to 30 farmers where they chat on WhatsApp and they share experiences, and sometimes they help each other work in the field. So farmer groups are really critical. And then we've also trained people from the Ministry of Agriculture who are acting as extension agents. And through those organizations, we're at the village level to train producers. We originally had this idea that each farmer would fend for herself and she would grow her own inoculum. But we did have some issues with contamination. It is important that the bucket that they're putting their rice in with the toothpicks and putting the lid on and sealing it up for 3 days, if that's not sanitized, then who knows what they're growing. They're growing whatever that was on their hands. So we really felt like we needed to manage that a little bit better, and the best way to do that was to train producers at the village level. And those producers, actually I think I have a photo, well, those producers then have this full system of manufacturing, and it's an economic development at the village level. So it provides revenue stream, keeps it at the village level rather than shipping in a chemical from the US or Germany or someplace. We tried to make it as streamlined as possible. We developed these bottles. This is basically like a Nalgene bottle with a coved lid, and the producer can put 6 of the wooden dowels that are inoculated with, this is the primary inoculum. They can put 6 dowels in that tube, and then they could cook the rice, put it into the sterilized bucket that's on the right here, and through that little black peg at the top, they can drop the 6 dowels in, and it's a very sanitary process. The cotton is for aeration, just so that as they're shaking it, that over 4 days, it has some breathing room. And this is a good system. This grows the fusarium really well. This is our team. That's Edelquin and Simon on the left, and they're training producers at the village level. Because of COVID, they're socially distanced by household, and they're learning how to do this so that they can then sell it to their farmer groups. And in the last year, we've done a lot of outreach, even though we don't have commercial registration yet. We've done a lot of demonstration plots. Over 600 demonstration plots got planted in the last 2 weeks in 4 counties in Western Kenya. And we've developed community-based organizations as partners, and we've had field days. And so it's been quite productive, even though we don't have the actual commercialization yet. We obviously, as a company, have set goals to increase the farmer crop yield, but we are a social impact organization, which means that we're sort of a hybrid between making money and being a sustainable nonprofit. So we're selling at cost, actually a little bit under cost right now, is what we have it priced at, so that as we reach scale, our organization will be able to survive without having to have more input of grant funding. And we're also working on some future aspects of distribution, which is a seed coating process, which would allow us to sell in a different venue than using the village producers. We would be able to sell in the small agrovet stores that are in each village. So we have our Kenya vision. It's pretty lofty, 250,000 farmers, but it's actually, we can account for those numbers. And then this isn't just an issue in Kenya. We have Striga, as that picture showed, the heavy infested countries, those were 18 countries. We ended up picking 12 countries out of those to focus on first. And then we actually have this bigger goal of shifting the whole bioherbicide market, with Africa leading the research. So to do that, we have created a Pan-African biocontrol science team. We trained these, all of these scientists at MSU in 2018, and they come from 12 different countries. We're actually going to add a scientist from Ethiopia in this coming year through a UNDP Cultivate program. All of them are early or mid-level career scientists. They have a different range of skill sets. Some of them are mycologists, some of them are have done commercialization projects. Some of them are focusing on cowpea, some millet, some striga, some sorghum. So we created this group to make a really strong team that can play with off of each other and build this innovation. So why biocontrol? This is sort of the big question. If you're looking at the commercialization, looking at the whole market, biocontrol has been growing steadily for the last 25 years. But biocontrol doesn't mean just bioherbicides, doesn't mean just weeds. It means getting rid of pests, it means getting rid of bad fungi. So that's where the entire market is. You can see that orange line, that is the weed, the bioherbicide market. It's flat line. There are no products on the market because it hasn't been successful. If you look at the synthetic herbicide industry, chemicals to kill weeds, that's over $35 billion a year is predicted by 2022. And there are a lot of reasons why that is ready for a shift away from the synthetic herbicides. You've all heard about Roundup, glyphosate. There are 14,000 lawsuits against glyphosate in 2019, and the organic market's growing faster. Weed resistance is a big problem. Weeds are learning how to adapt to the chemicals, and so now we're creating super weeds out of these weeds that are developing resistance. Like glyphosate originally would kill everything when it was first introduced in the 70s, and now there are 28 weeds that are resistant to glyphosate. So he likes to call it the perfect storm, that there's this buildup of issues against synthetic herbicides, and simultaneously there is a strength and a building team in the bioherbicide world. And Dad, you want to go through this, the pros and cons?
I think they're pretty obvious.
Yeah.
We have last century was a century of physics and chemistry. We didn't understand enough biology to really do what we're doing now. We're starting to figure it out. We're starting to figure out what's safe. We're starting to figure out that you need multigenic approaches. You don't just produce one amino acid or you will get disease resistance. So the main thing I think is that in this century, people like you will be able to find biological ways to do things that'll be safer and frankly more interesting. And that's what this course is about. You'll hear a lot of different lectures on a lot of different kinds of science. And maybe you will end up impacting the whole world the way it looks like Clara's doing here. So I'm a father, I'm very proud of my daughter. She's going for global impact. It won't be easy. There will be a lot of people against it. It takes true grit, and that's what we're after in this project. Now, striga only really exists in Africa and maybe a few counties in North Carolina where they brought it in accidentally. But there are at least 100 weeds, and 100 weeds that foul up waterways, or they, like poison ivy or so on. There are lots of weeds, there's lots of markets, and what we think is that we have a whole new industry, and we will find a way to put it out. So, are there questions?
I'll point out also that I majored in art history. I didn't have a science background at all, and I didn't work in science until I started doing some conservation work about 10 years ago and sort of eased into it. So your college degree doesn't necessarily dictate how you can use science in your career at some point. For me, I came from a nonprofit background and the impact background. And as much as I like and appreciate the science. I also know that if a farmer can increase her crop yield by 56%, that means that she'll have enough to feed her family. It means that she might have enough to sell some for surplus that she can sell for some cash that then she can pay for her kids' education. And if she can pay for her daughters to go to school, they can get past sixth grade. They have a chance of having fewer children, and they have a chance of getting married later, average of 4 years later, actually. And that shifts the entire perspective of what food security looks like for a continent. So the impact is really important. You have to connect the dots between what the purpose is. Are we just trying to kill weeds? Are we trying to just feed people? Are we trying to change entire systems? And so that's something that we're very, very cognizant of.
Questions?
This is our team. These are some of the funding and partners that we have as well.
Do you have anything else? Oh yeah, that's a good book. You might want to pick that one up. They don't even mention Striga. They should.
Yeah, so she asked if we've experienced any kickback from the synthetic competitors. No, he's presented at the biopesticide conferences. We're actually presenting again next week at the Biocontrol Africa conference. The big pesticide organizations attend all of these biopest conventions and workshops and meetings. They're keeping a close ear on what's going on. There's an alert in that industry that they need to find some other methods. When you're looking at a company, investors will always say, what's your exit? Where are you going to leave? How are we going to make our money back? And the most automatic exit would be to sell to Syngenta, Bayer bought Monsanto. Any of those, they're only like BASF, they're about 5 huge synthetic companies. And we're really hesitant to do that. There are pros and cons to if we were to do that. The pros would be that they have the huge dollars to push through regulation faster. They basically own, like in the US, they're at the top in DC running the systems. And so if they wanted to block us, they could. But I don't know, we haven't gotten that far yet. I think that they're keeping an eye on it. And at this point, we've been really generous in sharing the information with the African science team. It's difficult. There's a patent on the process, but that patent is up. So now it's just a matter of actually training and being in the lab and learning how to do the science. So it's still pretty limited, even though the patent has expired. And we do have a second training planned. Hopefully it got canceled. It was supposed to be in France in April, and that got canceled because of COVID. So our next big step is to train the African scientists. Eventually we'll have US company doing work here on, outside of MSU, doing work on domestic weeds.
We have a group of 300 organic farmers in Montana, and they passed the hat around and gave us enough money for 2 years research. And one thing they said is that they don't want this to go to a big company, because if it does, they will not be able to afford it. So it's a whole different business plan. You create a fungus that may live in the soil for 5 years. Monsanto and Bayer, when they deal with herbicides, they want to sell them every year. It's a whole different business model. Maybe some of you are business majors, and you'd be interested in how you put something like this together. But we do have a perfect storm with the chemical companies. We aren't sure that's the way to go, and we'll just have to see what happens. Any more questions? Well, I have a $5 bill that I guess goes to the one person that asked the question. Why do I do that? Because we have 240 students that are watching this
No, she's not. the one.
online.
She didn't ask the question.
Who did? You did. What do I know? Yeah. There's your $5. So thank you very much for attending. I hope you have some ideas. And I remember, and I will remind everybody that we have a final exam in this course, and that is have a dream. Money isn't the problem. Figure out what you're going to do to change the world. And this is just one little example. Thank you.